Okay, so test on Monday. We're good with that, right? Okay, then after that, we've got other stuff we're going to do. We're going to go over meiosis, and then we're also going to take a fake test on evolution. It's one of those ones we're required by the county to do. It's a pre-test. But let's start with this. And I gave you this system of these boxes. If you learn to do it this way, you'll never make a mistake. So they'll be really easy for you. So you look for the word dominant. And what's dominant here? It's a long hair. So we can write our first code. Big L equals long hair. Well, if big L equals long hair, what's going to equal short hair? Little l. Little l equals short hair. All right. Dominant, long-haired, homozygous. Homo means what? Same. So that just told me that what we've got here is big L, big L. Cat is crossed with a heterozygous. What does hetero mean? Different. Good guys. Different. Okay. So a heterozygous cat crossed with a homozygous cat. So let's do the cross. What goes here? What's big L, big L. It's homozygous. It has to be the same. This one is heterozygous. It's going to be big L, little l. We're done. We wrote the code. We know what our cross is. Now we go do the Punnett square. It doesn't matter where we put these. We just do them now. Big L gives us big L, big L. Big L gives us big L, big L. Big L, little l gives us big L, little l. Big L, little l. Cross is done. Now we just have to read what we did. So, let's drop down to genotypes. There's 3 genotypes. What are our genotypes? What are our combinations we can use here? Big L, big L. Big L, little l. Little l, little l. Okay. How many big L's do we have? 2. Numbers percent. Numbers percent. Numbers percent. We have 2. 2 quarters make how much? 2 quarters make how much? 50%. Okay. How many big L, little L's do we have?
2.
2. Again, 2 quarters give us how much? 50%. Okay, we're going to check the next one just like Nyla said. How many little L's do we have? 0 for 0%. We're almost done, guys. You guys are rocking this. Okay, phenotype. Geno. This has to do with the genes, the letters. We're going to look also that these are also called the alleles. Phenotype starts with a P, so does picture. So this is how it looks. All right, what are our 2 choices we're talking about? What are we looking at? Long hair? And, Kevin, what's the other one? Short hair. And you agree we could look at something and tell if it had long hair or short hair, couldn't we? We had Mike Gerbil back there. Could we look and see if it has really short hair or very long hair? We could look at that. That's the phenotype. Can we tell what its genotype is? No. We'd have to draw blood, do the whole thing to tell its genotype. So, long hair, Kev. How many long hairs do we have? If it has a big L in it, it's a long hair. We have big L, big L. We have big L. We have a big L. We have a big L. And we have a big L. How many do we have? 4. So long hair, we have 4. Short hair, how many short hairs do we have? How can we have more than 4? We have 4 squares. We have 0. Yeah, we don't have any.
Oh, I was
Yeah, you can't count this. Once you have a dominant gene or a dominant letter, a capital letter, it covers everything else up. So it only takes one big letter in any one of these boxes, and it's going to have the dominant look. The dominant gene is there. All right, you have a couple of things to do. Right down here at the bottom, I want you to write P1. P1. This stands for parent 1. Parent 1. This is our first breeding. This is our first mating. Okay? All right. So everybody's good here, right? Everybody can do this, right? Everybody can do this, right? I need to write. Okay, good. Because now what we're going to do is we're going to pick, let's do this cross. Okay, so we're going to take these two kittens and we're going to mate them. So go to your next blank page. It looks like this. And we're going to cross an L and an L, a big L and a little L. All right? Yes, you can put that in the problem. So you're going to do all the rest of it. I'm going to give you guys a few minutes, and we're going to come back, and we're going to walk through this one. Let's take a look at this. So, what's our code? Did our code change? No. So what is big L? Long hair. What's little L? Short. Our cross is up here. We gave us our cross. These are just two of the baby kittens that we're now breeding to see what offspring they will give us. So we've got big L, little L. We have big L, little L. So we have big L, big L, big L, little L, big L, little L, little L, little L. Here's what I want you to write now. Underneath this, put F1. This stands for family 1 or offspring 1. So this is family 1 or offspring 1. Okay? So this is just our first set of babies we have from that first mating. So let's do our genotypes. What are the 3 genotypes we can do?
Big L, big L, big L, little L, and little L, little L.
Perfect. Number percent, number percent, number percent. So let's go back up to our Punnett square where we're going to get this. How many big L's do we have?
3.
We have 1. 1. 25%. It has to look just like this. It's not 1. And it must, both letters in the box, I know, and that's where you're getting confused. So we got to make sure we know that. So it has to both be big L's. Is that the only space, Leandra, that there are 2?
Yeah.
Okay, good. Okay, let's go down here. How many little L, big L, little L's do we have now? 2. 1, 2. Good. So we have 2. What is the percentage then? 50%. Okay.
25%.
The top one's 25%. Remember, they got to add up to 100. Okay. And what is our last one? Little L, how many do we have? 1, which gives us what percentage? 25%. Now, let's go up here. I'm going to change colors too. If we have a capital L, it's going to have long hair. Hi. Does this have a capital L in this box? There's one that's long hair. Do we have a capital L in this box? There's one that has long hair. Okay, is there a capital L in this box? This has got long hair. Is there a capital L in this box? If it doesn't have long hair, what's it got to have? Short hair. It's our short hair. Okay? So now we go down to phenotype, how it looks. So we have 2 choices, long and short. How many longs do we have?
3.
3. which makes what kind of percentage? 75%, 3/4. How many shorts do we have? 1 for 25%, right guys? All right, here's the next thing we got to talk about. These letters, they're like different pennies. They're separate. We use them together, but when we break them apart, this is what it's called. One letter by itself is called allele. A-L-L-E-L-E. An allele. Write that down. Make that note. It's on your test. What is an allele? A-L-L-E-L-E. If we add both of them, they become alleles. Okay? They're alleles. If we put them both together. One by itself. Any one of these is an allele. A big A, a big T, a little t. That's an allele. Together, they form alleles. Remember when we were playing with the pennies? It could come up heads, it could come up tails. The other one could come up head, it could come up tails. That's what these are. This is heads, this is tails. Both of these are heads, both of these are tails. This is called this random assortment that Mendel talked about. Every time we flip the coins, it's random how we get these. It's predictably random, but it is random. So we can make predictions. It's going to work out mathematically, but it is random every time we do this. We good? We good? All right, here's your next thing. We're going to take 2 more of the cats, big L, and little l, little l. Find a blank that says the problem. Here's what you're going to do. You're going to do this crossed with this. I know. What you're going to do is there's one before when we started. Turn back, and it has stuff written in it, but the rest of it is blank. Use that one. Just ignore that part. I think it's about... Actually, we may have even used it. We did. We actually got through the doc. Do it on the back. Do it on the back of your paper. Just make a quick... You can follow the patterns you've been using. Make this cross. Okay, this is our, but here's what I want to show you. This is F2. What do you think F2 stands for? Family or offspring 2. So this is offspring 2. Or you could write 2 there. Offspring 2. So we're going to do this cross. Has our code changed? Has our code changed? No. So big L equals long. Little l equals short. Okay? Do the cross. We're going to do a big L and a little l and a little l. This is heterozygous, just to reinforce that. Heterozygous. This is homozygous. They're the same. Homozygous. All right? So now we're going to do our cross. And we find out that our genotypes is we end up with no big LLs. We end up with 2 big L little l's. And we end up with 2 little l little l's. So in this one, we do end up with 2 short kittens. So we've got long hair is 2, short hair is 2. And we're done. We usually carry it through, we can carry it through generations after generations after generation. If you can do that through 2 generations, I'm going to give you guys a round of applause. If you do it step by step, you'll be great. Okay, I'm going to give you guys a minute to finish that, we're going to work. So we're going to finish up our notes. And you can see this right now out in the senior courtyard. But let me put it this way. If I took a bucket of white paint, pure white, and a bucket of pure red paint, and I poured those into another bucket, what's the color of paint going to be?
Pink.
Pink. Thank you. If we take a red rose, we cross it with a white rose, we end up with a pink rose. This is because of what's called incomplete dominance. Neither one of them is strong enough to come out, so they share. When one gene for a certain trait is not completely dominant over the other gene, a blending, another word for blending is mixing, effect occurs, just like the paint I mentioned. Okay? You must know this. What is incomplete dominance? It's when one thing's mixed with another thing and we get something blended coming out. It happens a lot in plants. All right. Codominant shows both of the genes in places. So if we took a black chicken and we crossed it with a white chicken, we don't get a gray chicken. We get a chicken that's both white and black. The white says, I'm not giving up my spot. The black says, I'm not giving up my spot. So they share. If you've seen the big Jersey black and white milk cows that have black spots and white, this is what happens. The black says, I want my space. The white says, I want my space. They share. But they're both still black. They're both still white. They don't turn into gray. So this is codominant. Co means sharing. We good? Both those terms? Okay. All right. I want you to follow along in this. I want you to listen. I changed the letters here. So my letters here are different than your letters. This is easier. I'm going to tell you exactly what I want you to... I'm going to tell you exactly what I want you to remember for the test. A dihybrid cross for two genetic traits at one time. A dihybrid cross is a cross for two genetic traits at one time. All right. If you've had Ms. Moon's class, then you've heard about FOIL. FOIL stands for, you do this with polynomials, I think. First, outside, inside, last. So we're going to go right here. We're going to go to the first set. So we have a capital S, a capital D. First is done. Outside, we're going to go with the capital S, the little d. Inside, we're going to go with the little s and the big D. Last, we're going to go with the little s and the little d. All right? Does everybody see that? I don't expect you to do this. I expect you to follow along. You may see this on an SOL test. I've never seen you have to calculate it, but I have seen where you're going to see a question about what it is. Dihybrid cross. It's really more realistic, where we're mixing the color of puppies with their fur length. For example, a genetic cross for coat color and hair length in golden retrievers. If you haven't seen a golden retriever puppy, you've missed something. They are so cute. Many breeders choose the light coat color and long hair because many dog lovers prefer the puppy type. Short hair length, big S, is dominant over long hair, short S, length in dogs. A dark colored coat, D, is dominant over a light colored D coat. All right, so this is where we're crossing. So we're going to do FOIL on this side. So first, boom, boom, little s, little d. Outside, little s, little d. You guys should be writing this down. Inside, little s, little d. Outside, little s, little d. They're all the same because it's a homozygous match. Yeah.
You write down S and D?
Yeah, S and D, because we're fixing to use them.
Because ours, oh,
I know, I said use these, please.
okay.
That's what I said. Okay? Now we're going to just put them in like a Punnett square. A normal Punnett square has 4 squares. A die, how many does die? How many's die? How many's die? 2. Okay? So a dihybrid cross has 16 squares. We can do a trihybrid cross, like tricycle 3. It has 64 squares. We're just going to do this dihybrid cross. So it doesn't matter top or bottom where we put these. So we've got big S, and it's going right over here, big S, big D. Big S, little d. Little s, big D, little s, little d. These go across the top, and they're all the same. Little s, little d. Little s, little d, little s, little d. Again, because it's homozygous. Now we do the same thing we do on a Punnett square. The hard part of this is the only hard part, or figuring out what the traits are. Okay, but we're done. So now we have a big S and a little s. See this big S? See this little s? Just like we do in a normal Punnett square. We have a big D and a little d. We have a big S and a little s, a big D and a little d, a big S and a little s, and a big D and a little d, etc. Okay? Does everybody kind of see what's going on here? This is what you must remember. The definition of a dihybrid cross. We are crossing 2 traits. Eye color, fur length. Height against wrinkled peas, which is what Mendel did. Right? That part you have to remember. We're going to skip the mouses. All right. So many traits are controlled by multiple genes interacting with one another. I know you're going to write down the part that I need to tell you. Just this part is all you're going to need to write down. This type of trait is called a polygenetic trait. All right, so this is what the definition is. Polygenetic traits. Many traits are controlled by multiple genes interacting with one another. That's the part you got to know. Interacting with one another. That one line is all you need to know.
You write that?
Yep, that's all I want you to write. Polygenetic traits. Many traits are controlled by multiple genes interacting with one another. Because while we talk about height and weight and skin color and even eye color as being one thing, it's big T, it's little t. In real life, we're finding that more than one gene controls this. Is there anyone that is truly, let's say, black? No. There's shades of black. And this has to do with the amount of melanin in their skin. Is there anyone that's absolutely pure white? Well, actually, there is. And that's what we're going to talk about next, is albinos. William's not white. No, William's fair. He's not white. He's not pure white. He's not this white. It's not the white of this. All right. We good with this? Okay. This will be on Blackboard. This will be on YouTube. More than likely just YouTube. Okay, if you haven't finished this, I'm going to come back to this. Okay? So, which is no worries, we'll come back to it. Well, that's because it's weight. Look at the height of this guy. Or this girl, I mean. That's her coat. She's probably 6'2". Yeah, that's why she's playing basketball. All right. How much hair does this guy have? So the joking. He doesn't have any. Here's my question. First of all, what's this called? Baldness. How many women have you ever seen in your life that are bald?
One.
One? Out of how many women have you seen in your life? Very few. How many men have you seen that are bald? A lot. Okay. Write this down. Okay. Sex linked. Okay. Sex linked. Write this down. Sex linked. Huh? Just write it somewhere in these notes. You're going to have to be responsible for this.
We don't have that.
No, just write down sex linked. I mean, it's 2 words. Come on, guys. Sex linked. You can do this. I know you can. All right. Which side is the girls? Which side is the boys? This would be the right side.
That's girl.
Thank you. That's the left side. Okay, this is female. Make sure you know that that symbol means female. Make sure you know that this symbol means male. All right. N stands for non-baldness. B stands for baldness. Non-baldness is dominant. Okay? So if non-baldness is dominant, is this female bald? No, because this non-baldness dominant gene canceled this baldness out, right? We good? Males have, okay, how many chromosomes do we have? You guys ought to know this fast. 46, thank you. No, we don't, Nyla, we have 46. We have 46 chromosomes. Those are somatic. Those are skin cell chromosomes in our body. How many do our gametes have? 23 each. Where do we get 23, the first 23? No, we got one from 23 from our mom. Where do we get the other 23? From our dad. All right. On the 23rd chromosome, that's where we find on the karyotype what sex we are. Okay. So, what do you notice if this is a chromosome of a female and this is a chromosome of a male? Other than the color, what do you notice the difference between those 2 chromosomes? What did you say, William? Which one is shorter, male or females? The males. Do you notice that? What does it not have on it any place? A letter of any kind, right? It's neither non-bald or bald. All right. So we're going to make them have a baby. You're going to tell me if that offspring is going to be bald or not bald, okay? First baby. Bald or non-bald? Not bald, okay? Everybody can play. The rest of you guys are just sitting there being quiet. Okay. Another baby. Bald? Non-bald? Bald or non-bald?
Non-bald.
Non-bald. Why?
The end is stronger.
Good.
That's it.
It's dominant. All right? Good. Now, bald or non-bald?
Non-bald.
Non-bald.
Dominant beast.
There's no B. No B anywhere in it. Okay? That gene doesn't exist in this combination. Now, bald or non-bald? Why? Because there's nothing here to counteract. There's not another gene here. The male chromosome is smaller than the female chromosome. Mainly, guys, all we deliver is what the sex of the baby is going to be. Inside that egg is all the DNA, all the coding, everything else for pretty much the entire baby. We deliver what it's going to be. We deliver that by our genes. All right, this is called a sex link. Now, going back, why is it called sex linked? Think about the exercise we just did. Why is it called sex linked? Who controls whether you're bald or not? Wrong, not your parents. Who controls? Which one?
The male.
Does the male have anything to say about it? Does he carry? If males could just develop by mitosis, and we never saw a female, would we ever have a bald male?
No.
Why? You're right. Why? What's missing? The B. There is no bald gene here. Males have nothing to say about it. Do you see that? Until the female brings in this bald gene, there would never be any bald males. If this gene did not exist in females, there would be no bald males. Okay? So this is sex linked. It means this gene travels through one or the other of the parent. Most of them through the female. And not that the females are bad, It's because males don't have this other part of the chromosome. So let's say that do you know what albinism? You're an albino, you have no pigment. Let's say right here, down below this bald gene, was an albino gene. Now males don't carry the albino gene. Okay? What happens now? What happens to the offspring of this child? He's albino. That's how sex-linked traits work. And mainly it has to do because we are missing so much of this gene to counteract the other part. Does that make sense? Okay. Moving on. All right. I want to get to one. Okay, mutation. A mutation is a change in a gene. Mutations can occur in a single gene or an entire chromosome. They can be caused by conditions of, in fact, let's get you to write this down. Or copy it off of Blackboard or off of YouTube when you do this. You need to know this. It will be on your test in a form. And it will be in the SOL. Main thing you could see them asking you, the SOL guys, is what are some conditions? They can be environmental, they can be X-rays, ultraviolet radiation, radiation from nuclear energy. This is what happened at Chernobyl when the Russians had the reactor that went critical. They have generations of babies now that are born without arms and born without legs because of these mutations. There's a whole book called The Chernobyl Babies. And it's about all of this bad stuff that happened being exposed to radiation. All right. All right, Kevin. We're going to work on this. Here you go. We don't have this. I want you guys to watch along with me. Okay? You have to tell me as we go along here what we're going to put. In squash plants, yellow fruit is dominant. There's our word. What are we going to do with the word dominant? Take it, do this. What are we talking about? The fruit. What color is the fruit? Yellow. What letter are we going to make it then?
Y.
What kind of Y? Okay, so we can write our code starting right here. Capital Y equals what? Yellow. Now we got to find out what is the other part. Okay, to white fruit. Oh, then what is little y, Kevin? White. Okay, so far we got some good stuff here. If two plants heterozygous, hetero means what? Come on, guys, you ought to be able to roll this right off your tongues. Hetero means what? Different. Different. So now what it's saying is we're making a cross with these, and it's two difference. So we have a big Y, a little y, a big Y, and a little y. There you go. It says, what are the possible genotypes? Well, okay, so let's do our genotypes. What are our three combinations we could have? What are the three combinations we could have, guys? Big Y, big Y. Big Y, little y. Little y, little y. So, how many big Y's do we have? Well, we do have one. Good. How many big Y's, little y's do we have? Two. Do we have any little y's? Yeah, how many? One. Okay, so we have all three of them, right? Big Y, big Y, big Y, little y only? Is that the right answer? No. Is it big Y, big Y, big Y, little y, and little y only? Really? I got a big Y, I got a big Y, I got a little y, don't I? Okay, let's leave that one in the running. Big Y, little y only? No. Big Y, little y, little y? No. There's our answer. All right? Okay. Here's what we're going to do now. I have a bunch of the definitions done for you. They're loaded into the smartboard. But I would like for you guys to work on those notes that you picked up today, those vocabulary words. I'm going to put them up here. You need to write those down. What I'm going to do is go very quickly through them with you and write down just the simple words. You need to go back through, look at them, and write the complete definition down. Consider that your homework. So let's go to here. Okay, it says genetics test review. Homozygous, homo, same. Big A, little a, little a. Heterozygous, different. Big A, little a. Dominant. Dominant means big A covers, hides little a. Recessive. Recessive is covered or hidden unless it's homozygous. Homozygous. Then it's going to show. Genotype, that's big A, big A. Big A, little a, little a, little a. Phenotype, how it looks. Blue eyes. Tall. Long hair are all examples. Okay? Alleles, an alternative form of a gene. These are the letters. This is an allele, is big B, and big B, little b is alleles. Plural. It's the allele. It's the visual representation of that gene. Mendel, father of modern genetics, he was a monk who came up with the Punnett square. Actually, they came up with the Punnett square later, but we give him credit for it. And he worked with peas. Over 10,000 of them. Okay? Hybrid, offspring, big H, little h. Have you ever heard of a hybrid car? What's a hybrid car? What's it do? What's unique about it? Hybrid, come on, Kev, you're a mechanic. What's different about a hybrid? What can it run on? Okay, some they think it means it runs on gas and electricity. Okay, electricity. Makes it a hybrid. It's different. Two things that are different. Are these two different? Yeah. When we talk about different, they're different sizes. This is a heterozygous. Purebred. Another word for homozygous. You talk about purebreds when we're talking about trying to keep a bloodline pure coming down. We're raising horses or dogs or cats. Okay. Karyotype. Remember I showed you the chart, and it's in your notes, of all the chromosomes. And we said the 23rd one over here was the sex chromosome. It is a snapshot of an organism's genetic makeup. Okay, makeup. Homologous. A homologous pair is we're talking about two chromosomes. And the thing about this, just like what does homo mean? What does homo mean? Thank you. In this homologous pair of chromosomes, they have to be exactly alike. And what they will do on the test is sometimes they'll put like and they'll say, are those two the same? And even though they have the same structure, look at the gene placement. They are not the same. They have to be exactly the same. And that includes the number, location of the genes. Punnett square, we know what that is. P1, parent 1, we know what that is now. F1, family 1, first offspring. F2, family 2, second offspring. Chromosome 23, males. Remember, this symbol is for male. This symbol is for female. Law of segregation. This says that all of this happens randomly. This is one of Mendel's rules. Randomly reform pairs during fusion of gametes. Just remember, it happens randomly. Do not get overly hung up on this. Diploid. Do get hung up on this. How much does di? Di? How many is 2? 2 sets of chromosomes. Okay? So, somatic. How many somatic cells do we have in our bodies? 46. Diploid. Diploid. Di. And here's the 23. 2 times 23 gives you what? 46. 23, one set inherited from each parent. 23 from each parent. Okay? Haploid. These are the sex cells. Okay, let's review sex cells. What do males have? What's the gametes in males? Oh, come on, guys. Thank you. This is sperm. Okay, female. What is the gamete for a female? An egg. In humans, 23 chromosomes each. Meiosis. You can take meiosis out. Okay? Gametes you're going to have to know. Crossing over, you can take out. Okay, here we go. Right-handedness is dominant to left-handedness. Kevin, where's dominant? Did you see it?
Yeah.
Right-handed, okay, so there's our dominant. We start looking for what is dominant. What is dominant? Right-handedness is dominant.
Right.
What's our letter going to be? Big R equals right hand. Okay? To left-handedness, what is our little r going to be? Left. Okay? Suppose there's a couple which are both right-handed. Okay, so we have a couple. We have a male and a female. They're both right-handed. What are our genotypes that could go with both of these? If they're right-handed, what do they have to have? What do they have to have? A big R, right? Okay, right-handed, right? Okay, so would this make this dad right-handed? Would this make this dad right-handed?
Yeah.
Okay, would this make this dad right-handed?
No.
Does it have a big R?
Yeah.
Then he's going to be right-handed. Okay, mom, would this make mom right-handed?
Okay.
Would this make mom right-handed? Okay, so those are the combinations we can have. We're not sure where to go. The mother has a left-handed mother. Okay, so she has to be left-handed. If for a recessive trait to show, what does it have to be? If mother's left-handed, the mother has a left-handed mom. So this lady, this mom, has a left-handed. The only way she can be left-handed is little r. Little r. I know, guys, this is boring, but you got to do it. Right? The father has a mother who is also left-handed. And they're doing this for a reason. Okay? If this little r is going to show up, this is what dad is. This is what mom is. Okay, so plug these in. Okay. It says, do the Punnett square, do the genotypes and phenotypes. I think you can do that. I get upset when I'm writing a whole bunch on the board, and nobody else is writing anything, Saeed. Like all the little quick notes I just gave you to remember. I do hope you review this. All right. We're going to do one more. One cat carries a hetero. Hetero, same or different? Different. One cat carries heterozygous long-haired traits. Oh, they even give it to us. SS. Okay? And its mate carries homozygous short-haired traits, little s, little s. So they're saying big S is long-haired, little s is short-haired. It is short. Use the Punnett square to determine the probability of one of the offspring having long hair. Walk in the park. We do a Punnett square. We've got big S, little s. We got little s, little s. We have big S, little s, little s, little s, big S, little s, little s, little s. Okay? So, how many big S's, big S do we have? Really? I don't see one. Are both of these capitals? Are both of these capitals? Then we don't have any. Guys, you're going to have to be careful on this. Now, how many big S, little s's do we have? Good, 2. And how many little s's do we have now? 2. Okay, so this would be 50%. This would be 50%. Okay, so they're asking to determine the probability of offspring having long hair. What's the percentage? Really? How in the world did we get 100% a heterozygous long-haired, big S, big S? What are these guys going to be? They're short hair. So we have 50%. Okay.
Big phones.